Hello everyone, today we will discuss about non-ribosomal pathway. Non-ribosomal pathway. So before going to the non-ribosomal pathway, we will first discuss about the ribosomal pathway and then we will get back to non-ribosomal pathway, and we will also compare how far this non-ribosomal pathway is different from the ribosomal pathway. So let us discuss first about the ribosomal pathway. Ribosomal pathway. So this ribosomal pathway is nothing but the translation process. Translation process which is actually found in most of the cells. Found in most of the living cells. Most of the living cells. Maybe of prokaryotes or maybe an eukaryotic cell. And if you see the different you say components which are, or the requirements which are there in the ribosomal pathway, or particularly in the translation process, that is, you say ribosome, which is the first one and very essential, which provides the site for the protein synthesis. This is first requirement. Second, we know about the mRNA, that is messenger RNA, which carries the message particularly for the synthesis of a particular type of protein or peptide. So carries message for the production of a particular type of protein or peptide. Particular protein, or you can say peptide. Okay. And in addition to these two essential components, some other components like you say amino acids, okay, which will be joined together to form these polypeptides or peptide chains. There are different type of amino acids, as you know. Then tRNA is also there, which will transport this amino acid. So transfer the amino acids to the site of protein synthesis. Protein synthesis. So this is the requirement, and another one more requirement is also there, that is certain protein factors. I am writing here only protein factors. If you have studied briefly about the protein synthesis process, then there is also three fundamental steps are there, like you say initiation, where certain initiation factors are involved, and polypeptide chain formation, and the last one, some protein factors are also involved in the termination process. Termination is a process, and some proteins are also there which are involved in the modification. Okay, some factors are also involved involved for the structural modification. Okay, so this is the scenario of, you say, ribosomal pathway. So in case of, you say, non-ribosomal pathway, there will be no involvement of a ribosome. There will be no involvement of a ribosome and mRNA. Amino acids, these are involved here, mRNA, but the specific type of amino acids, that is non-proteogenic amino acids, will involve here. We'll discuss it later. Ribosome and mRNA. So there will be no involvement of this ribosome and mRNA. You can say the independent. So it is independent of these two things, like ribosome and mRNA. So if someone wants to define the non-ribosomal pathway, how you will define this one? If we go for definition, so it is the pathway through which non-ribosomal peptides are synthesized. Through the enzyme complex called as non-ribosomal peptide synthase enzyme. Which is independent of ribosome and mRNA, as written here, no involvement. So when you say non-ribosomal peptide, is there any difference between non-ribosomal peptide and the normal peptide? Yes, there will be some difference. Like non-ribosomal peptides, okay, are those peptides which do not have, you say, proteogenic amino acids. So like in the ribosomal peptide, they usually contains proteogenic amino acid. Here it contains non-proteogenic amino acids. Non-proteogenic amino acids. Non-proteogenic amino acids. When you say non-proteogenic amino acids, so these non-proteogenic amino acids are, if you take some example, then like your D amino acids. You know about this D, desterotatory amino acid. That is that represents the spatial arrangement or spatial arrangement of elements in a molecule which has the capability to rotate the plane polarized light into right and left. So based on that, actually you can decide whether it is desterotatory or whether it is laboratory. So when it is said to be the desterotatory, it will write to the left. When it is because it is come from the Greek word dextro, okay, which means the left. So left form of, you say, amino acids, depending upon the structure. If you take some example also, like you can take D-alanine, D-alanine. You can take D-glutamic acid, D-glutamic acid. Okay, and usually this you say amino acids are so-called as non-proteinogenic amino acids. That means that usually do not involve in the protein synthesis process. If you take some more, you can take GABA. And besides the GABA, you can also take beta-alanine also, alanine. Besides that, you can take citrulline. You know, citrulline and ornithine. These two are, you say, particularly found in the urea cycle, because the ornithine is the, you say, the compound, or particular the amino acid which are found in the urea cycle are so called as ornithine cycle. Okay, so ornithine is the most essential component in case of, you say, urea cycle. And if you see the urea cycle, this ornithine is actually the first, you say, the last, you say, amino acid which are to be converted, okay, in the urea cycle. Okay, if you remember the urea cycle. Under the urea cycle. Okay, so this is what you say, you can write the non-ribosomal peptide. There are the few examples you can take here for the D amino acids. Then as we are discussing about the non-ribosomal peptide which contains the D amino acids, so apart from that, structurally this NRP or non-ribosomal peptide, non-ribosomal peptide, in short NRP. This structurally, particularly can be available in two forms. One is, you say, chain form, or you can say linear form or branched form. Chain form, I can say, which is very branched, or in other words you can say branched form. Another one is that is a cyclic form. If you take the example of chain form, it's a long chain form. If you take some example of chain form, that is Vibrio Bactin, and for the cyclosporine is the example of cyclic form, cyclosporine. Okay, and here also you can take also example for singolysine. Is another example. So usually this non-proteogenic amino acids which are involved for the production of non-ribosomal peptides, these are usually found in case of, you say, bacteria and, you say, fungi. These are found in these NRP is found in fungi and bacteria. In multicellular also they are found, but that is because of, you say, the presence of these bacteria. So in multicellular organism, these peptides are found only because of these bacteria and fungi, which are serving as a symbionts. And if you see the structure, like you say cyclic form in the chain form. In the chain form, it is heavily branched. I have said here heavily branched. Why it is heavily branched? Due to the addition of different type of you say reactions. So for the different type of reaction, like you say uh, N-methylation, this is one process through which the methyl groups are to be attached. Addition of, you say, methyl group single bond CH3. Second is that you say N-formyl methyl, N-formyl methylation, like addition of single bond CHO, like formaldehyde HCHO, or you can say CHO aldehyde addition. And glycosylated also another process, glycosylated, and another one is acylated, halogenated, and you can take also hydroxylated, in case of you say glycosylated, there will be addition of carbohydrate addition. Acylated, like addition of, you say, acyl groups, like R single bond C double bond O. This is the acylated group or acyl groups, you can say. Okay, halogenated, addition of, you say, halogen elements, addition of halogen elements. You know about halogen elements? If you see the periodic table, you will find in group 17 or VIIa, halogen elements are there. Like you say, if you take an example of chlorine, bromine, iodine, astatine, etc. Okay, these are the halogenated, you say, element when it is attached to any type of structure, so called halogenated process. Hydroxylated, this represents the addition of a single bond OS. You know this one, hydroxyl group. So this is how this chain form is branched by the addition of different processes. And next to it, if you go for the cyclic again of different types, like you say different type of cyclic. I'm writing here the cyclic one, the second one, cyclic form. So in the cyclic form, you will find two you say type of cyclic form. One is you say oxazo oxidines, sorry, oxazodines, oxidines, oxidines. Another one is you say thiazolines, oxazodines, thiazolines, oxazodines, thiazolines. When you say oxazodines, these two are oxazodines and thiazolines. These two are you say, you can say five, you say member of heterocyclic ring compounds, you say aromatic compounds. These two are you say of five member heterocyclic compounds. Okay, if this structure particularly contained oxygen and nitrogen in its structure, like here, if you go for like this one, if this is the structure, one ring, if the N is present here, then it contains O and N. That's why this is called, you say, thiazolines. And here you will get the sulfur containing ring. That's why it is called, as you say, thiazolines. Sulfur and also the nitrogen. So if sulfur and nitrogen is here in the ring structure, then you can say thiazolines. And if the O and N is there, then it will be called, you say, particularly oxazodines. Okay, so this is the two different forms of using non-ribosomal peptides. I think you all clear about the non-ribosomal peptides and non-ribosomal pathways. So we will discuss next about the non-ribosomal peptide synthase enzyme, how it will, you say, produce the non-ribosomal peptide, and what are the processes of these non-ribosomal peptides. Thank you.